Here we present an automated system based on optical coherence tomography, or OCT. This allows us to monitor biofilm structure over large spatial scales or extended periods of time. OCT imaging is well suited to resolve structures in the micrometer range. However, it's currently limited to a maximum area of about 250 square millimeters. Biofilms often exceed this scale, especially when the differentiation is driven by large-scale environmental gradients.
The experimental setup allows us to monitor the three-dimensional morphogenesis of biofilms over large spatial scales and extended periods of time. The system is fast, precise, and it works autonomously.
We study the morphogenesis of biofilms in streams where they drive important ecosystem processes. However, the system may be used to study biofilms in other natural system or engineered environments.
The software for positioning, image acquisition, and analysis is written in Python. They are available through Jupyter Notebooks. These are user-friendly, freely available, and flexible solutions.
We believe that the visual representation of the setup helps other users to reproduce the installation and to better understand the software. We hope this inspires other researchers to adopt similar approaches.
Here is an overview of the installation. The system is composed of a precision positioning device, the OCT probe, and it is assembled around a plexiglass flume. Begin by wiring the positioning device by following along with instructions posted on GitHub. Once connected, install the GRBL server as described in a separate GitLab page.